This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 476, recorded on January 12th, 2018. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today here in New York City, Dickson Despommier.
Hello, Vincent.
And I will just give a brief weather report. It is cloudy, currently 15 Celsius.
15.
And all the snow is melting. And, you know, this polar wave is gone.
Yeah, for a time being.
Let me also introduce our guest, since he's right here and he won't have to wait. We have an in-studio special guest today. He's come all the way from Salt Lake City, Nels Elde.
Hey, Vincent, great to be back.
Welcome to TWiV.
Great to be here. Back-to-back podcasting festival.
We just did
today.
a TWiVo.
Outstanding.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And it's also 15 Celsius here, 59 Fahrenheit, and it is pouring rain.
Really?
It's just dumping down, and the snow's melting really fast. And I'm sure we're fortunate in that we're on sort of a rise on the street, but I'm sure there are parts of town where we've got lots of flooding.
Yeah. Well.
Yeah.
From southeastern Michigan, Kathy Spindler.
Hi, everybody. I want to tell you guys that we had 56 Fahrenheit here yesterday, at least, which is 13 C. And then this morning we got colder temperatures and snow and ice and stuff.
Oh, gee whiz.
Wow.
And right now, let's see, it's 24 Fahrenheit, -4 Celsius.
That's probably on our way.
Yeah.
And from Austin, Texas, Rich Condit.
Hi there, everybody. It's 51 Fahrenheit, 11 C. It was warmer. It's getting colder. It's going to freeze tonight.
It was warmer.
So, yes, it's coming your way. Oh, and it's nice, bright, sunny, clear blue skies. So it's good.
We have some cold temperatures and snow coming here, right, Dickson?
We do.
All right. That's it for the trout fishing then, right?
Yep. We can dream.
So Nels is visiting
Go ice
on a seminar visit, right, at Nels?
Yeah, that's right. So I spent yesterday at NYU and was hosted by Victor Torres, a microbiologist in the microbiology department there. Really fun day with both virologists and bacteriologists.
Well, thank you for making this part of your visit. It's really nice of you.
Thanks for having me. It's really fun to sit
and
in.
Anyone else who visits New York and
Consider it your second home.
stop by and do a podcast, right?
Great.
Yeah, you can come by anytime. We have quite a few things for your listening pleasure today. So don't touch that dial.
Don't go away.
Who used to say that?
That's a good question. Whose show was that? Jack Penny's?
I don't know.
Interstitial?
I just got Kathy Spindler's email about the ASV this week. What's going on, Kathy?
Well, first of all, you're going to get a postscript, a PS email, because even though we looked at it numerous times with at least 3 sets of eyes, we left out one of the 6 satellites. We left out the one about microbiome interaction with viruses. So you'll get a PS sometime in the next day or 2 or 3. But that email to members has all kinds of good information. You can also go to the asv.org site or the meeting site itself, asv2018.umd.edu, to learn all about the July 14th, 18th, 2018 ASV meeting. I want to tell you that 18 days from now, or from when the TWiV is released on Sunday, is when abstracts are due and when travel award applications are due. So in addition to students and postdocs being able to apply for travel awards, teachers of undergraduate virology are eligible to apply for travel awards, and they don't have to be members and they don't have to submit an abstract. Also, Latin American scholars can apply. They don't have to be members, but they do have to submit an abstract and present something. So those 2 other categories are something we haven't really talked about in the past, but there are travel awards for those kinds of people.
Cool.
January 7th in the Washington Post Online, an article by Aaron Blakemore entitled "Viruses Have a Big Impact on Our Lives, and This Podcast Gives Them Their Due."
Could that be us?
It was This
Who
Week
was
in
it?
Virology.
Yes.
Ever ask yourself what's the latest on viruses? Don't blame yourselves if you haven't. Enter This Week in Virology. Yeah.
Cool.
And I think this is great. Thank you, Aaron, for doing this. And lots of people found out about TWiV as a consequence, including the director of communications here at Columbia. Said, "Cool, I didn't know you did this. Why not? Why not?
Never a prophet in your own. Never a prophet in your own land. Never."
Right.
Congrats to you all. Washington Post stamp of approval.
Did we get a bump on this? Did we get a bump?
Did we get a
Oh, that's a good question. Let's look. Let's go to Libsyn, and we can look very quickly to see if we got it. And we should thank Jeff Bezos as well, right? Because he owns the
Post, right?
and Aaron
he's probably had a few viral infections in his life as well.
I hope Jeff Bezos is not taking that active a role in the editorial content.
He said he wouldn't. He said he wouldn't. Yeah.
No, you signed that to Rupert Murdoch.
Right.
The statistics site is down, so I'll check
Fox
back
News.
later. I'll check later. I wanted to do that before the show and get it lined up. Libsyn is our hoster of our files, and they are down.
Right.
Well, they probably crashed because of all the additional traffic that was coming in.
Exactly.
You bet. Well, the website, microbe.tv, crashes every morning at about 3 a.m., and I get an email about it. And it's saying, we've had to restart your server. I don't know why. I still haven't. I don't have time to figure it out. But I have to do that one day. I don't know. It's probably robots crawling the site, you know.
Right.
Yeah.
All right. Kevin finally claimed his book from a few weeks ago. That was Viruses by Kevin Cordingley. Let me see what Kevin said. Greetings, TWiV. Sorry for the late response. December's always a bit hectic, and I've been getting ready for grad school, proposed to my fiance, etc. Really? Those are good excuses?
No.
And I've only gotten around to listening today. Don't know if it's too late to claim it, but here's my information. So Kevin is from Massachusetts, Alan.
Oh, cool.
Yep. So I got his address. So I will send Kevin the book. Meanwhile, not a single person has sent in a completed crossword puzzle to win a copy of Neurotropic Viruses. So no one wants to do some work to win a free book, huh? Or maybe they don't like neurotropic viruses.
Or they don't like crosswords?
It could be.
Wow.
I know it was a hard crossword because a lot of the clues had to do with ASV 2012, which, like, you know, what was this biotech exhibitor at ASV 2012? Who the hell remembers that?
Yeah.
You have to get the other ones to get that one.
And, well, yeah, you can always do that with crosswords, right? You don't get every one.
That's right.
All right. But the idea of using a crossword was submitted by Islam Hussein, and I went to the website that he suggested, and I made crosswords for the new edition of Principles of Virology. We're going to put crossword puzzles in.
Nice idea.
For each chapter, and it turns out to be pretty easy to do, and maybe make it a little fun. So,
cool.
Thank you, Islam.
Very nicely done.
We have follow-up from Mark. Dear Dr. KAV, you attributed the science of HIV listener pick to me in episode 475. I can't take credit for this. Maybe you mixed up emails. Yes, I did. So Brian was the one who picked that, who was an assistant professor at UMass. He picked Janet Iwasa's Science of HIV site. Nels, she's your colleague there over at the University of Utah.
Yeah, great choice, Brian.
We've talked about her multiple times, and we have to get her on TWiV.
Great idea. Days. Yeah. Another colleague along these lines at Utah, Mariah Meyer, just had a book released on visualizing scientific data,
nice.
Which is worth taking a peek at. I'll try to find that link.
Okay. Send us a link. Alan, can you take the next one?
Sure. Anthony writes, thank you for reading this on TWiV 475. And this was the Cuban scientist travel ban. Dr. Henderson, D.A. Henderson, I assume, noted that at a time when both the Soviet Union and the United States were openly preparing for the possibility of destroying each other, he worked with the Soviet public health officials to effect the eradication of smallpox. How can one explain in today's environment of emerging disease, and when Cuba poses not even a semblance of a threat, that U.S. scientists will be restricted from traveling there? Government employees must respect the whims of a fool. The winds and the tides will not.
All right.
Yes.
Thank you, Anthony. We can't say it here, but you can say it.
Oh, we could say it here.
It looks like Nels has a new manuscript on bioRxiv. What's that about, Nels?
Yeah, so I posted this earlier this week, hoping for a little of this real-time peer review transparent feedback, knowing that I would be a in-house guest on TWiV today. I was pretty excited about that. No, so this paper has been brewing for a while in our group, or this manuscript. And so this is a continuation of some earlier work on this idea that poxviruses, large DNA viruses, adapt through recombination-driven mechanisms that cause gene duplication events to sort of almost a breathable genome, or this idea of the genome accordion, as it's been a little bit, to my chagrin, described, coined by us, and then it's sort of taken on a little bit of a life of its own. But so anyway, we've extended the experiment of evolution, where we passage the virus in replicate populations. The sort of new spin on this one is that we took a virus that has a expanded accordion, if you will, extra copies of a gene, which is an adaptive maneuver for the virus. But there's also a single point mutation in that gene that starts to spread through the population as well. And so we had the question of, well, what happens when you have two different adaptive mechanisms sort of filtering through the population at the same time? Do these things cooperate? Do they compete? Is it additive, synergistic? And so we just did the experiment and let the virus tell us. And so the trick here, though, was in order to type the viruses or understand what was happening with the evolution, because there are all these copies of the gene, sometimes up to 20 copies of the gene, we needed a way to phase or type these things to be able to say, in copy 1, does it have the point mutation? In copy 2, yes or no, on and on. And so we went to this Oxford nanopore, this relatively new sequencing technology, which is really fun. It sort of democratizes sequencing. So you have the device, plug it into your laptop. They've got a new iPhone version that's coming out soon for field work. It's really fun.
It's small. Like a harmonica, right?
Yeah, exactly. And
rather than an accordion.
It's all, this is what I mean. This is the
downsizing.
That's right. So anyway, we used that new technology to type these viruses, and we were able to get the magical thing, not only sort of the being able to do this with your laptop, but you also get really long reads. So in our hands, the longest read we've gotten is north of 100 kb. And I think
amazing.
Yeah.
And what do you get with, MiSeq type sequencing?
Right. So Illumina based stuff like MiSeq, you're, between 150 and 300 base pairs, if you're doing pair N. So this is a huge step forward. There's other long read technology, PacBio. That's a sort of competing technology. The difference there is it's not the size of a harmonica. It's the size of like a refrigerator.
That's amazing.
And they are expensive contraptions. So these are sort of, the price point is really getting attractive.
So this little harmonica-sized device, how much does that cost?
So we do it a little bit in bulk, but it's not super cheap. We're doing about $500 a run using it.
Can you reuse it?
No. So the flow cells are that's the consumable of it is the flow cell, and that's disposable basically. So that's one run. So,
and then you do the reactions, and then you load them into this flow cell.
Exactly. You just take a P1000
This thing is less than 6 inches long, and it resolves all of this. And, we used to pour these 3-foot long gels to resolve DNA sequences. Amazing.
Shark's tooth comb gel.
Yes.
Yep.
Yeah.
Oh, yeah.
Mm-hmm. Yep.
And we didn't get that many bases on it either.
Right. So, yeah, we just in one pop can, go to your laptop and pull out 100 kb. So we did that. And what we saw was kind of cool. So if you start to count or follow sort of the descent of that point mutation in the arrays of genes or the copies of the genes, it quickly homogenizes, meaning that if at copy position, depending on the frequency in the overall population as the experiment unfolds, if it's 50% frequency in the whole population, then as you go through, half at each position in the array will have that allele. And so sort of it's really kind of, it's really striking when you see this. So if you just simulate,
there's a puppet master
or something is happening. We don't know exactly. We're trying to get onto that, but somehow the point mutation is just filtering evenly through the populations. And so if you simulate it, and we did that at different sort of bottlenecks or effective population sizes to simulate what was happening in the real experiment, what you see with a random distribution is an enrichment in the low copy viruses. So the genomes that only have 1 or 2 copies should start to get that point mutation. And the ones with many copies will sort of suffer. There'll be a sort of a desert of the point mutation. And yet, because it's not random, something is happening where it's homogenizing. We think the outcome is that the virus will hold on, or as a population, you'll have more of the viruses that have more copies. So to go back to the half-baked accordion analogy, you basically hold the accordion open before you contract it back down or squeeze it back down.
That's known as the Lawrence Welk effect. I don't know if you knew that.
Thanks, Dickson. I'm
glad we never used the accordion concept for a title or anything.
Right. We use squeeze box, right?
Yes, I know.
Listen, do at any time all of the copies acquire the mutation?
Yeah.
100%.
So we've run the experiment for 20 what we call passages. So each passage is a 48-hour infection, or
cell culture.
Right? And so the first paper was passage 1 through 10. This paper is passage 10 through 20. And so the cool...
Okay, I see your plan here.
Hey, wait a minute. Hey,
I know what the next paper is going to be.
I'm not going to give that away. Stay tuned. But
we've heard that song.
But so here's the
it's
cool...
like Richard Lenski, right?
Yeah, there you go. Right. Yeah, he's been doing this for 20 plus years, and he's at passage 50,000 or something like this. But so we... if we go back and actually look at the frequency of that point mutation in the population, so at passage 5, it's at about 10% frequency. And then actually for the next 5 passages, it's on this weird plateau, basically, where it stays at 10%. While now in the population, all of this recombination-driven action is still happening. So the accordion, so to speak, is expanding. But then the magic sort of happens between passage 10 and 20, where you go from 10% frequency of that point mutation up to 95%. And so getting to your question, Vincent, it's not at 100 yet, but it's getting really close. And so
at some point, is the accordion going to collapse and you'll have few copies?
So that's the question. If you hit 100,
that's paper number 3.
we're waiting.
To be announced.
To be announced. I don't know if or when the accordion will collapse. So certainly, and this came up on TWiV 474, which I just listened to with great interest. Call me Fish Mule. Call me Fishmael. That's bridge too far for me on the title. But
will the circle be broken? That's what we want to know.
Will the circle be broken? So, like that massive giant virus, BSV, Bodo Saltans virus, super interesting giant virus.
The Arnold virus.
That's right. And so the idea from Curtis Suttle and colleagues there up in Vancouver, right, was that this thing has hundreds of copies of a gene.
Yeah, it's a
right.
What's that? It was a
ankyrin repeat
protein.
Right.
Got it.
And so, taking that maybe as a hint, it could be that we're in for a long ride here before we would see a contraction. Now, certainly, if you take the virus and put it in a different cell line, where you relax the selective pressure on having extra copies of that gene to defeat this immune defense that the virus is sort of competing against, then immediately that accordion collapses. And reaches a new equilibrium. And so it's almost like a counting mechanism for how much selection is there in what we think is a trade-off.
So if you don't use it, you'll lose it.
Right.
So Dickson, last time you had difficulty understanding why it would lose it because what's the problem with extra DNA? Remember you said that? Or are you going to deny it now?
No, I won't deny that. But upon reflection,
upon further reflection,
that's right. Upon further review, the play on the field stands.
Okay.
So, but it's a interesting and open question. Actually, is what are those sort of how much contraction is there? But I think at least from this paper, this part of the work, the hint we're getting is that, and the surprise for me really was that you might hold it open longer. Because as a consequence, whatever the mechanism of this homogenization. Yeah. So,
hey, Rich Condit, are you there?
Okay. Yeah, I'm here.
Seriously, this is vaccinia, and you're so quiet.
I've been waiting.
Okay.
Up to this point, he's taking notes.
I only have 50 questions. So, the story in my head that I came away with from the earlier papers was that under selective pressure, you would first amplify these genes to give you extra copies to play with. Okay. And then with those extra copies, you could experiment with you being the virus. This is, of course, a conscious virus. You could come up with some mutations that might be beneficial. And once you had a couple of those that worked okay, then you could maybe forget about the extra copies and collapse it if you wanted.
So before you started editing, you saved an extra copy for archival purposes.
Exactly.
Yeah.
But what I come away with from this paper, check me if I'm right, is that a lot of your in the lower passage guys, you had a lot of single copy guys with the advantageous mutation, as if that amplification was not a prerequisite for making that. Okay, it's sort of contrary to the original model. Am I seeing this correctly?
Yeah, right. And so we draw a new model or a revised model that separates out the two. So we now, by using this new technology, the Oxford nanopore sequencing, and comparing it to Illumina and going a little bit higher resolution, we're not 100%, but getting closer to the idea, or having evidence for the idea that perhaps it was the point mutation that showed up and then it got into those arrays and then homogenized, as opposed to the original model. I don't think this is mutually exclusive, by the way, where the copy number goes first, then the mutation shows up, just like you're describing it, Rich. And so what we're saying now, or what we're observing, is that perhaps both are possible.
So just as an aside, this is a little sort of larger teaching moment. I want the audience out there to appreciate that one's own models and hypotheses of what go on can change with new data.
Yes.
And it's okay. Everybody's okay with this. That's fine. Yes. Okay. You don't have to stick to the idea you came up with to start. Are there politicians
even if it
listening
was a
there?
Cool idea?
Even if it was a cool idea. Okay.
Is Dr. Mark listening?
Number two. What, can I remember this? What number two was? No, I can't. Damn. It went away. I'll get it. It'll be back.
Another teaching moment for me is, I've always been intrigued over the simplicity of evolutionary process, in the sense that the environment always changes. Mm-hmm. There are random mutations, and the random mutants that are best fitted for the new environment survive, and the others don't. And in this case, the environment is in the immune cell. As the cell develops immunity, the virus responds with random mutations. At one point, it has a gene now that says, that's no longer valid. I can reproduce inside of this now thoroughly immune cell. And then the more copies of that gene they acquire, the less likely it is that they'll be wiped out.
Correct. And so when we talk about the random mutations here, they're kind of come in two flavors. The one that most virologists are familiar with, or the way we think about most, just a single letter change, a point mutation. These DNA viruses also do these recombination-driven mechanisms, those extra copies of the gene. And the combination of those two the dynamics turn out to be pretty interesting. And just to say, so thank you, Rich. I'm first of all relieved that for the positive teaching moment. But I wouldn't I'm not trying to dismiss the idea of the accordion, just revise it. I think it's getting, the deeper we go, the more texture we're getting, or the more details, the more interesting and sort of complicated it's getting.
So when you withdraw the immune attack on the virus, the accordion disappears.
It does. So and it depends. It's interesting because you can remove.
Thank you, boys.
You can remove that pressure in different ways. So if you pick a different cell type that lacks that immune block, then that relaxes the selective pressure. And what we see is almost immediately, and this was in that first paper from a few years ago, and thank you again for covering it on TWiV. I remember that episode. It was, my heart was in my throat with excitement at the time. It was so fun to have that on.
I hope you didn't choke on it.
Well, we'll see. Not so far so good. But we did that experiment in the 2012 paper. And in fact, when we moved to that other cell, it collapsed down. We could recover viruses with much lower copy numbers, or even just a single copy with that point mutation. And so the new revision here, what I think it's telling us is that there's a mechanism somehow in place to actually hold the accordion open longer. And that's this homogenization, which then takes away one of the trade-offs, which is that you would quickly contract if you just have that single copy and it's enriched there. But what we're seeing is it's distributed through the arrays. And so we think that might, something like that giant virus that you were just talking about 2 episodes on TWiV that has 150 copies, perhaps if there's a similar mechanism of gene homogenization, that might explain those massive collections of extra copy numbers in a virus like that.
So do you think this happened originally in your original experiments, but since you weren't doing the sequencing, you didn't see the propagation of that point mutation?
Yeah, it turns out it's really tricky. So that first mutation to track exactly in the population where it was. So we used both both Illumina and then PCR-based methods last time. And then we added in the Oxford nanopore. And it was right around the 4th, 5th, 6th passage when the action happened. And it was almost simultaneous. And so our best guess is that it actually showed up at a single copy and then quickly got into the arrays. But I think that the idea still holds, that if you have, for example, 100 copies, you're 100 times more likely to hit on a point mutation like that. And so hopefully, I mean, we're continuing to test and probe and let the viruses tell us, but I think they're telling us some really interesting things about virus evolution using the pox virus model with all these great tools. Amazing history, great work from folks like Rich Condit, who's here with us, and but trying to see whether some of these rules might apply to things like these crazy, giant, ocean-going viruses that might have a lot to say about carbon capture and distribution in ecosystems right under our nose.
So there was another couple of things that interested me. One was this was one of these moments in reading a paper where, okay, so you're passaging this virus, okay, and there's the potential, depending on how you passage. This goes back to the von Magnus effect. Potential when you passage, if you're passaging in a relatively high multiplicity, you can carry through the passages viruses that by themselves would be sick, okay, but are complemented by other viruses. And I forget quite how this came up in my head as I was reading the paper, and I was thinking to myself, man, I wonder what multiplicity he used, and he really ought to do different multiplicities. And bingo, a couple of pages later, you're doing different multiplicities. So I really appreciated that. And you said that your original multiplicity of infection was like at 0.1 or something like that.
Correct, that's right.
And then I started to, and I think that was your highest multiplicity of infection. And I started to wonder, I wonder if the particle infectivity ratio, I've always wondered about this, if the particle infectivity ratio is 10, so you've got 9 dead particles around and 1 infectious particle. I've always wondered whether those 9 dead guys, some of them could carry, otherwise inactive genomes that could be complemented by an active genome. So, when you're doing a multiplicity of infection of 0.1 PFU per cell, you're actually doing 1 particle per cell, and you're going to have some double infections. So, but at any rate, you went way back to a really low multiplicity, and it didn't make any difference. Okay, so this seems to be multiplicity independent. I appreciate that, a subtlety, I suppose, for the not cognoscenti out there.
No,
what if all the viruses were dead? What happens to the DNA? Doesn't that just get digested then?
No, it's a great question. So first of all, thanks, Rich. And we did, as you're saying, the experiment over 4 logs of MOI. So we start, our lowest was 0.001, and then the highest was 1. So 10-fold more than the.
Oh, you went all the way up to 1.
Okay. To 1. Yep. But then, and the phenomenon sort of holds across that 4 orders of magnitude of start, which I think is kind of fascinating. And we're kind of poking on that. But then, Dickson, getting to this idea of the defective interfering particles and other systems that have been studied for a really long time. Really interesting questions. Whether these things can complement, how long do they hang out in the populations? Does this influence the infections, both perhaps augment them or even interfere with them in different ways? And so I was just talking to Elde Gaydon at NYU yesterday, who's really thinking about this idea of defective particles in influenza infections. And they have some really cool results where basically when you look at each of the 8 segments and you do PCR across the 2 or 3 kb, that you have what she calls these Batman year effects. And that means that as they look at the reads, deep reads, they start piling up at the edges of the segment. And then you see a hollowing out or a lower coverage in the middle. And so what they're doing is they're collecting all of these segments that have deleted out or are missing parts of the genes, in some cases the polymerases, and just have sort of the non-negotiable ends of the segments that persist somehow in these infections. And so they're starting to think about this idea with
they're called viromirs, by the way.
Viromir. There you go.
I just made
she has got Batman years.
So, like
yeah, right. So anyway, I think it's a great question. There's so much interesting stuff happening as we're sort of teasing out at a genomic level how these populations of viruses infect and spread.
So this is
40 years ago, I did serial passage of vaccinia to see if anything like the multiplicity, anything like a von Magnus effect showed up. And I didn't do it rigorously, but I didn't see anything like it. So as far as I can tell, any sort of weirdness that goes on is going to have to be revealed by exactly the kind of stuff that you do, Nels. Mm-hmm. It's not going to show up in the typical fashion.
Mm-hmm. Mm-hmm.
When I was at Penn, Carolina Lopez talked about similar studies looking for renewed interest in defective interfering particles of influenza virus. It's very interesting because those were forgotten for years, and now people have gotten interested again.
Yeah, I agree. She's doing incredible work on it too. Yeah.
So this has been submitted, I presume, right?
Not yet. So currently it's just out on the bioarchive.
So you're waiting for Cell to ask you.
No. We've invested some time and energy in this. It's been a couple of years. It's actually a really fun collaboration with Genomics, Genome Science Lab in my department. This is Aaron Quinlan's lab. And so we both teamed up a grad student on the project. We came at it from the virology side, and then they came at it because they were interested in the sort of recombination, copy number variation in genomic data sets. And so it's been sort of a 2 or 3 year conversation between our two labs and the two grad students in particular, where they've both been sort of cross-training. So anyway, we got it up. Part of the reason to get it, I mean, one of the great things about the bioarchive, right, is that you can kind of go on the record and get it out there. Hopefully get some, constructive criticism from these great friends and peers.
Certainly gotten a lot of tweets.
Yeah, that's right. So kind of try to distribute it. But then it's sort of a living, breathing document, hopefully, where we can continue to revise, take some of those into account. We also wanted to get it on the records. We have a grant application coming up. And so just to show that we've delivered maybe on some of our promises from before. Right. So we try to renew something. And then finally we'll revise and then submit it, but we're still open and seeing.
You can expand and contract different sections.
Uh-oh.
That's right.
That's right.
Accordion in and accordion out.
Good. So one last thing, Nels. I got the impression, I just checking myself again, towards the end of this, I got the impression that the point mutation and the duplications have essentially an additive effect. Is that correct?
Agreed.
You get better, say, resistance to the immunity if you've got both around than you do with either one alone.
That's correct. And we tested the as best we could. We isolated the mutations from each other to test the fitness consequences in this cell infection system. And so if you just have the point mutation, you get a little bump in fitness. If you just have extra copies, you get about an equivalent bump. And then if you put the two together, it's as you're saying, Rich, an additive situation as far as we can tell.
Right. Okay, cool. Excellent.
Yeah. Thank you.
Nice.
Keep on passaging, dude.
Keep on passing.
We're on the case. Thank you all.
So this snippet I picked because you were going to be here, Nels. Thought you might be interested. It's published in PLOS Pathogens, The Paradox of Hepatitis B Virus Evolution as Revealed from a 16th Century Mummy. And the authors are Zoe Patterson-Ross, Jennifer Klunk, Gino Fornaciari, Valentina Giuffra, Sebastian Duchenne, Anna Dugan, Debbie Poinar, Mark Douglas, John Sebastian Eden, Eddie Holmes, and Hendrik Poinar from University of Sydney, McMaster University, University of Pisa, University of Melbourne, and the Canadian Institute for Advanced Research. And that's it for that. That's our snippet.
It's a cool one. Check.
Yes. Okay, we mentioned it.
Yeah.
So this is an interesting story where they sampled remains of a child mummy that was found in the sacristy of the Basilica di San Domenico Maggiore in Napoli, Italia.
It just rolls off the tongue.
It was exhumed between 83 and 85 and had been previously studied. It's about 439 years old.
It's 1983 and 1985.
That's correct. And at the time when it was originally exhumed, they did an autopsy and saved some samples, and then they reburied it. But the mummy seemed to have a rash on it, a vesiculopustular rash, which looked like it might be smallpox. And electron microscopy revealed some particles that could be pox virus-like. So then they revisited it in this current study, did some sequencing. And the sequencing reveals no vaccinia virus or pox virus DNA. However, so it reveals hepatitis B virus in multiple organs, skin of various places, bone, etc. clearly hepatitis B virus. They could assemble an entire genome, and they could say exactly what type it is among all the different genotypes that have diversified. They could clearly show that it was ancient and not a contaminant by the frequency of cytosine deamination, which in an old sample typically occurs at the ends of DNA molecules, whereas in your lifetime it happens throughout your DNA. So if it were a contaminant, that would show up, and they could tell that. However, the puzzle here is that when they do a phylogenetic analysis of this sequence with all the Hep B sequences from all over the world for the past 50 years, it looks like a contemporary genome.
Yeah, wild. Which is why you would want to rule out the contamination, right?
And they made a point of saying it's not a contaminant because they went through all this trouble to do that. And there are many other things.
Maybe it's a fake mummy.
No, even looked at that. They did mitochondrial DNA sequencing, and it clearly is the right place. And, the clothing is of the right time and so forth. I think all the provenance is good. So that's where then they say, well, probably what happened is that this virus entered humans way before 1500 and diversified a long time ago into all the genotypes that are currently circulating. And although we know that the genome undergoes mutation, that must be purged away over time because there are constraints in the genome that limit mutation. So we don't really see from 460 years ago to now, we don't see much variation such that you could say this is a contemporary isolate and it's an old one. What do you think, Nels? Does that make sense?
Yeah. Well, so first of all, as a poxvirus enthusiast, a little disappointed that it wasn't the actual pox scenario. But no, so I think it's this fascinating paradox, actually, right? Just as you're describing it, Vincent, where we know that modern day viruses are rife with mutations, right? This is the whole idea. Hepatitis B virus currently today, there are these clouds or swarms of diversity if you recover them from a patient, for example. So how is it then that you could have the same thing, but that long ago, if the thing is changing so fast? Right. And we've seen a few examples of this now. So I'm thinking there's another really cool example, I think human herpes virus 6 or something like that, where if we go into our genomes, there's not a 0% chance that we might have an entire herpes virus integrated into our chromosome. And so and by the way, very similar situation where it looks like a modern herpes virus. And so I think it's exactly what you're saying, Vincent, which is that we sometimes underestimate the constraints on viruses. We kind of give them too much credit for moving because of how it looks in that really sort of close view. But when we step back, it sort of reveals how these things are actually remain the same over really long periods of time. So anyway, I think it's super fascinating. I remember seeing this a few weeks ago and thinking, very cool.
In this case, they do point out that HBV has a lot of overlapping open reading frames.
Yes, correct.
And that could pose some serious constraints because that sort of there's many fewer opportunities to introduce silent mutations. Okay,
right.
Because most of the mutations are going to have an impact and would be purified. This same group did do a poxvirus. Are you familiar with this? Nels, we covered this. I can't find what episode we did it on. I don't think I'm making this up. I have the paper.
Yeah,
I'm
no,
pretty
you're
sure we covered it.
Yeah. No, 100% right.
17th century variola virus reveals a recent history of smallpox. And if I understand this correctly, that was almost the opposite situation, where what they came up with was that there was a lot more variation, or the variation was much more recent than they might have expected. And out of that paper, they drew the conclusion that maybe smallpox as a human pathogen is a much more recent phenomenon than people had previously suspected.
That's right. I remember that. We did do that for sure.
Yeah, another cool example. And in that case, so first of all, I think there are a lot of the possibility to continue sampling. So in these mummies, it's a pretty conspicuous thing to have a pox mark, right? And so it's sort of you can see that target. So super cool. And then, I think that might be giving us a hint actually about a difference in sort of evolutionary styles, if you will, between the smaller RNA viruses and the larger DNA viruses, where the point mutation rate is sort of lower, which is sort of, again, paradoxical for the reversal we see here. And yet a lot of the evolution there involves, recombination driven mechanisms, as we were just talking about, or even the capture of host genes, where the viruses are constantly sampling and then sort of repurposing these genes in a sense to then modify or dampen the immune response. And so I don't know the answer. I just think it's really interesting and worth continuing to push on. But these viruses have really different sort of adaptive styles, and I think that might be sort of unfolding before our eyes as we take these samples back in time. So yeah, really cool.
I was sort of, when we did that paper, I was sort of shocked by the notion that smallpox might have been as recent. I mean, I'm not, frankly, based on historical records, buying that it's as recent as 16th or 17th century, because it's hard to believe that the diseases that were spread by the conquistadors in the 16th century, well, still 16th century, 14th, 15th century, weren't smallpox. Okay. But it's possible that it's, the older ones, they were variolating in China in like the 2nd century AD, I think. So something's funny here.
Agree. I think the jury is still out, and
yeah, okay.
I think we need some more data points, some more, and yep.
That bit of that. Actually, my first reaction to this was that the conclusions they came up with based on the molecular biology from that Variola mummy paper were really at odds, it seemed to me, with the historical record. Even though they kind of blow off the historical record in that paper and say, well, that's kind of anecdotal and stuff. There's some pretty robust history there. And then I was wondering, well, maybe. Although the problem is it's sort of flipped. It's opposite. Maybe the clock is wrong, that the clock that they're using in that paper is wrong too.
I think almost certainly. And actually, so in evolutionary studies, whether it's virology, evolutionary virology, or evolution of, say, our own species, the dates are constantly being sort of proposed and refined or blown out of the water. This just happened actually recently with some work in the last week or two that came out. Talking about indigenous populations that made that transition from Asia across the Bering Strait.
That's exactly right.
Yep.
Exactly
and that new data sort of revised or blew out of the water a recent idea that set the timing of that back almost 100,000 years. And so we're still, as scientists, arguing about these sort of fundamental things. And so the use of molecular clocks, I think we'll actually get to this as our episode unfolds in another kind of situation, where sort of the timing of mutations is really tough and fuzzy, and the assumptions that we make to try to put times to these processes can really, can fall apart pretty quickly as new data comes into view.
You mean
one message that I'm getting from this is that one virus's molecular clock can be very different than another.
Yes.
I mean, HBV with overlapping reading frames and a small genome size really has much different constraints than something like vaccinia with a huge genome, potentially, these recombination driven events and more room to exchange information with the host, et cetera. It's going to be different.
I want to go ahead, Kathy.
Well, I want to jump in. I'm not sure when there's a good point, but for the listeners who might be wondering about the fact that they found pock-like things on this mummy. So that's what made them think probably at first that it was smallpox. And this is an open access journal. You can look at the pictures, and on the face and some bone, the arm bone, you can see these pocks. And so the whole time I was reading the paper, I was like, okay, it's HBV, but what explains
what explains that? Yeah.
And finally they get to the end, and maybe this explains some other pock-like things along the way in history, but they say that there's a Giannotti-Crosty syndrome that's a rare clinical outcome of HBV that presents as a papular acrodermatitis in children between 2 and 6 years old. Now, granted, I didn't look up what papular acrodermatitis is, but it sure sounds like it's explaining what the pox were.
Sure.
So that I thought was interesting. And also, there still is not an explanation for a finding that was not mentioned. Originally with this mummy, they did protein A gold complex immunostaining for pox virus, and some other researchers, and there was a positive result. So that positive result, maybe just wasn't controlled well enough, maybe some kind of false positive. So that part has not been explained away, but perhaps the appearance of these pock-like things on the mummy has an explanation, and that may explain other pock-like things throughout history. And
also kind of along a similar line, since this is a mummy, they don't have any internal organs.
Right.
So they can't, not that they'd be in great condition in any case, but they can't look at the liver or anything else to try and get at a diagnosis that way.
Yeah. And that brings up another question I had, which was, how were they, were these locations where they were finding the virus, did they make sense for hepatitis B virus? Because they didn't look at the liver, but the positive places they found it were in the femur, muscles, the frontoparietal bone with skin, and the thoracic skin. And then they do describe somewhere later, I'm not sure if I'm going to be able to find it, that the virus can also be found in other places, not just in bone. Oh yeah, that the virus is found in mononuclear cells, which are protected by the hydroxylate matrix of the bone, and so that could explain why they found virus in the bone.
Mm-hmm.
Right. Yeah, I think this is probably all related to just viremia.
Possibly, yeah. Rich, by the way, variolation was practiced in China from the 15th century.
Okay, I had that wrong.
So that's more in line with the
That's more in line with
timing,
what they're saying.
yeah.
So another question then is, what is the earliest known positive smallpox DNA detection? How far back in history had they gone and detected it?
It may be this paper.
Yeah, this could most likely DNA, yeah.
So in that sense,
that was 17th century.
Okay.
And by the way, I searched the TWiV archives and Googled around, and I didn't find the 17th century one. I did find TWiV 187, the mummy, which is the 16th century Korean mummy with hepatitis B that's also referenced in this paper.
Yeah.
Right.
Yeah, I think I agree with Rich. We did do that.
So there's a ton of mummies in a very specific location, in the Middle East, that I'm surprised as heck that someone hasn't done that study, or maybe they have. I don't know. But what viral infections did the ancient Egyptians have?
And there's the classic one that's quoted as a mummy from something like the 3rd century BC. Yeah. That has a smallpox-looking lesion on it, and that's one of the standard things that's used to establish that smallpox has been around for that long. But as we've just learned, that's not necessarily rigorous.
So it could have jumped from animal resources or animal sources into humans.
Well, most likely did, for sure.
Yeah.
But
when
is the question.
So, in other words, in summarizing, work on mummies is not necessarily a rap.
Oh.
Right.
Mm-hmm.
By the way, I found the link to the mummies, the original investigation of the mummies that I put in the.
Yes.
Yeah, that's cool.
That's a fun read. I read that whole
yeah.
thing. It's really,
I just skimmed it, but they talk about the fact that they found some rooms, 3 at least, which were certainly intended for the dehydration of the corpses. They were equipped with sand beds for collecting the cadaveric sewage, and with wide ventilation shafts.
I'm sure. By the way, the Egyptian mummies come with urns in which all of the organs are present.
All the organs are stored.
Right. So you can look at all of the organs if you want.
Yeah. Yeah. Although you got to trust that it's from the right mummy. So Nels, one last thought on this purging. We did a paper not too long ago showing that, when a mosquito picks up West Nile virus, huge diversity, and then the next bird host purges it all.
Yeah, the bottlenecking effect. Yeah. Super interesting from sort of a population genetics.
Yeah.
Yep. I think I would just close by saying I think this snippet also highlights maybe a shout out actually to the adventurous virologists who go after these ancient samples, because that's a high wire act. I mean, it's easy to sit in the comfort of a podcast and say, "Oh, why didn't you get this or that?" But
you call this comfortable?
So far, so good.
You got scotch in front of you.
But that's really tough to get. As you pointed out, Vincent, to get the to know that it's not a contaminant. And we're talking about trace amounts of nucleic acids, which are, and to just get this at all, it's really fun.
This is a whole,
and you can't, if you screw something up, you probably can't get more samples.
No, exactly right. This is a whole science in itself, ancient DNA analysis. You have to have a facility just dedicated to that. You can't have any other DNA in there.
That's right.
And that, you need
except the people that are working.
In fact, in this paper, they said the McMaster ancient DNA facility was used for this. So there's a whole science that has emerged to do this so that you minimize contamination, for sure.
Yeah, that plus the next generation sequencing, to me, were the two technical breakthroughs that came together that allow this kind of study.
Right.
All right. We have a paper which is also very interesting, and it broaches a topic we really haven't touched on, I believe. I could be wrong. And it was published in Science, and it is entitled An Interferon-Independent LNC, long non-coding RNA, promotes viral replication by modulating cellular metabolism.
Right.
By Pin Wang, Yunfang Xu, Zhuzhou Wang, and Shuitao Cao from the Second Military Medical University in Shanghai, the Zhejiang University in Hangzhou, and the Peking Union Medical College in Beijing. And the idea here is, the first sentence of the abstract, viruses regulate host metabolic networks to improve their survival. Just think, a virus infects its cell, starts to churn out tons of genomes, proteins, needs energy to do all this. It's got to crank up the thermostat.
Damn right.
And we are just beginning to realize how this happens. And in fact, in the last edition of our textbook, Principles of Virology, we included a chapter called The Infected Cell, which in part talks about what we know about how viruses turn up the thermostat, make more energy in cells in order to facilitate their replication. And that's what this paper is about. So that's why I thought it was cool, because it's really a topic that we haven't talked about very much. Here, and there's some, as I mentioned, we have a chapter in the book. So there's some information on what viruses can do to, or how viruses turn up metabolism. But for the most part, the aren't really known. And they stumbled on one in this paper.
Up to now, it's been mostly phenomenology, I think, right?
Yeah, the mechanisms.
The viruses do this.
Exactly.
Right.
So I believe they stumbled on it because they were looking at long non-coding RNAs. And we've talked about those before. In particular, one of them, which is produced from an endogenous retroviral genome that seems to regulate pluripotency in mammalian embryos. But there are lots of these, thousands of these in mammalian genomes. And what do they do? People are trying to figure it out. So in this study, they were trying to sort out, I think it's the relationship of these long non-coding RNAs with interferon. They were looking at long non-coding RNAs, which I'll call link RNAs, okay? Shorten it, in both wild-type and interferon receptor-deficient macrophages with or without vesicular stomatitis virus infection. So what's the effect of knocking out interferon receptors on the production of these link RNAs? So they found in this study a group of virus-induced but interferon-independent link RNAs. They're induced by virus infection, but doesn't matter if interferon receptor is there or not. And that's probably not what they were looking for. I bet they were not. And they said, whoa, we should probably look at these. And to their credit, they did, which is good.
Right, because the interferon-induced ones you kind of expect. That's part of the cell's response to the virus. But one that's induced but isn't related to interferon, what's going on there?
Yeah, I mean, the interferon-induced ones are interesting because you'd like to know how they work.
Sure.
How are they antiviral, right? And they're, as the name implies, they are non-coding. You can steal my pen?
No, I'll give it back.
Okay. All right, so then they say, what's the role of this in VSV infection? And they can knock down these link RNAs using RNA interference. And if they knock down a specific one, which has the nice name link RNA ACOD1, aconitate decarboxylase 1, that's the nearest gene to this link RNA.
I thought of it as ACOD1, but then I looked Massachusetts.
ACOD1 is good. And you could make a title up.
He will. Don't encourage him, please.
So if they knock down this link RNA, virus titer went down. Knock down link RNA, virus titer goes down. So right away you're thinking, wow, this must be good for virus replication. So this is called link RNA ACOD. What did you call it? ACOD1.
Okay.
It can be stimulated by many different kinds of viruses. Why did you? Oh, this is a title for the episode. Okay, show it to Nels. What did we look at here? Many different types of viruses. Does anyone remember what the many different types were?
Seven.
In the supplementary data. And by the way, this paper has a record supplementary data file. There are 26. There are 6 figures in the paper and 26 in the supplementary data file.
Which are 3
That's
more
not
papers.
A supplement. That's the paper.
Those are 3.
It's amazing.
That's right.
Sendai virus, VSV, HSV, herpes simplex, vaccinia virus.
All right, good. Thank you very much.
And they did a bunch of different cell types as well. I mean, the minute they said VSV and macrophages, I said, give me a break. We got a cow virus in macrophages of mice. You got to do more than that.
No, they just wanted to milk their data for all it was
Rich
worth.
says, give me a break. I love it.
Also, the 6 figures that are in the paper all have at least 5 or more parts. Yeah.
Yes.
All right. So the point here is that it's induced by infection in many cells and organs infected with a variety of viruses. So it's not just VSV, as Rich says.
Right.
Give me a break. No, it's actually seems to be broadly important.
That's right.
And they also showed that this lacks coding potential. They did some translational experiments, ribosomal profiling data.
This is incredible. The whole thing is incredible. What a journey.
Ribosomal profiling. Yeah. Remember that?
Oh, yeah.
I was, we were
that's Joan Stites on steroids.
We had a textbook meeting, and I said, should we put sucrose gradients back in the methods? And everyone said, yeah, everyone's doing them again. We took it out because it disappeared, but now it's back. Okay, so what is regulating the production of this? So we know it doesn't require interferon receptors. They also showed it doesn't require IRF3, which is an important transcription factor needed for the production of interferon. So it's independent of that, but it is dependent on NF-kappa B. A complex, it's a transcription factor complex. I don't know, probably does everything, right?
Yeah, it does everything. I was thinking you tweak NF-kappa B and it's like calling 911 or, pulling a fire alarm.
Yeah.
And so then they went on and showed 3 binding sites. There's a subunit of NF-kappa B called REL-A, 3 binding sites for REL-A in the promoter for this link RNA.
And REL-A is part of NF-kappa B.
NF-kappa B. So the idea being part of NF-kappa B is binding the promoter of this link RNA, and that's involved in its production.
So we don't know how the virus turns on NF-kappa B. Okay,
No.
so that is a missing link. But from then on, we're okay.
Yes. You want another 20 supplemental figures, right?
Yeah, right.
But they did study that induction, and that by doing reporters and so forth, and that they showed that those motifs are important for the activity of the promoter using luciferase reporters.
And there's only 4 people on this paper.
Yeah,
I mean it's incredible.
I kept thinking as I was reading through this thing, wait a second, this is a boatload of work.
Yeah.
Yeah.
And it's all good.
They then ask, what is the role in virus infection? They knock it down, and it reduces the production of VSV in macrophages. They also looked at herpes simplex type 1 and vaccinia virus, similar reductions by knocking down link ACOD1, or what is it? ACOD1, yes.
ACOD1.
No effect on viral entry, but probably working at some stage of viral replication after entry. They have mice now that lack the gene for link RNA ACOD1. And VSV in those mice is substantially reduced. And when you challenge these mice with a lethal dose of VSV, they all survive. And
I have a question. Those mice that were knocked down, that limited the viral replication. Did they grow slower also?
The mice?
Yeah.
They didn't say anything about the mice.
Because these are metabolic stimulation without a virus.
Well, but there's probably something else, right? There's never one thing, right?
Well, yeah.
That's what Nels and I concluded on TWiV.
No, I would agree, but the question is then, did this have some metabolic effect on the host as well?
Yeah, it's a great question.
Yeah.
And one I think you could address, actually. So there are a few labs that have, so it can be, exposing maybe a subtle effect can be tricky, especially maybe in a coddled laboratory environment, maybe not as comfortable as a podcasting environment. But, but then there are some great systems. So these like mouse barn systems, or even like outdoor enclosures where you could start to take knockout mice, compete them basically over a few generations
in real time,
environment against their otherwise genetically
with or without a hawk.
Yeah, right. Exactly. Yep. And then come back a few generations later and say, who are the kids? Who has the most kids? And then that might scratch that itch.
They do have a bunch of microarray analysis and otherwise in this paper, where as part of those experiments, they compare wild type versus the linked knockout mice. And from what I can see, there's no obvious differences, in gene expression profiles in those. And later on, do they do, metabolome as well? There's not a big difference there either. So knocking out this link RNA on the host in normal circumstances, the whatever effects there might be are subtle. So this is as it turns out, this is going to be a regulator, but a fairly subtle regulator. It's not something big time. So you'd need your barn experiment to do it. So the big things show up. The big differences show up in plus and minus infection.
So Haverol.
That guy talked about that on the TWiV we did at Utah. Remember the barn experiment?
Exactly. This is my colleague Wayne Potts.
I'm curious as to why
Wayne Potts. Heck, he was at Florida.
Yeah, he knew. He said he knew you. Yeah.
Oh.
I'm raising my hand now to get attention.
Too bad.
Sorry.
Welcome to my world.
Yeah, thank you.
Yeah. Well, at least I can see his hand.
You can do that. That's true. So these are disparate viruses. There is nothing in common with any of them. Is that true? And yet they're all doing the same thing.
Yeah, this seems to be a wide
what mechanism. Do they have? They've got to have something in common.
They're viruses.
No. Not all the viruses did that. This group did, but the whole bunch of others didn't.
They must all have to turn up metabolism.
Not always, right?
Tweaking NF-kappa B is not an uncommon phenomenon in a virus infection. As a matter of fact, some of that may even be the host's attempt at some sort of antiviral thing as well.
That's right.
Okay.
That's right.
Yeah, they came in, they set off the alarm.
This is not even the best part, but there's more. Let me just say that
there's more
in humans.
Oh yeah.
This a similar RNA is upregulated in response to flu infection, and if you knock it down in human alveolar basal epithelial cells, you interfere with flu replication. So maybe it's important in people as well. In mice, IRF3 and the interferon receptor are not involved in its regulation. There's no effect on apoptosis and autophagy. And here is an important thing. The RNA, this link RNA, is located in the cytoplasm. Okay, so now what is it doing? They did microarray transcriptome analysis to look at what deficiency does on other metabolism-related genes. And what they found is that metabolic pathways are the ones most significantly affected by the loss of this link RNA. I don't know, maybe it would be good to turn down your metabolism, right, Dickson?
I take a little bit of that.
They did whole metabolomic surveys using liquid chromatography and mass spec.
Good Lord.
It's a lot of work.
So
in cells with and without this link RNA, and you compare metabolomics, the small molecules that are produced.
That was my question.
And they found that infection leads to large changes in wild-type cells, which are abolished by link RNA deficiency in these metabolomic, the small molecules, the solutes and so forth that are products of metabolism.
So infection is acting through link ACOD1 to induce a major change in metabolism.
That's right.
Right.
So next, and that's not even the best part.
No.
They said, what's interacting with link ACOD1?
Right.
So they purified from cells link RNA ACOD1.
This is where things start to get kind of ridiculous.
And they say what proteins are stuck on it.
Oh.
They separate the proteins by gel electrophoresis and identify them by mass spec. And that is like that one sentence. It's really hard to do that.
Yeah, they aren't fooling around either. When they go after this link RNA protein complex, they've got a mixture of 45 biotinylated primers to decorate the RNA with and pull it out with its protein on it.
Lord.
And it paid off because the difference between plus and minus ribonuclease is a single striking band on this.
Yeah. This, Rich, reminds me of your friend from Buffalo who made it big with C-gas. Remind me of
right.
Guy.
James Chen.
Similar kinds of amazing.
Yeah, it's a heroic effort.
Every sentence is like months and months of work.
Now, there's also a lot of technological advancement. I mean, mass spec has come leaps and bounds in the past 10, 20 years, but this is still, it's a boatload of work.
A binding protein that they identified is glutamic oxaloacetic transaminase. Got to go.
Yep. A
key metabolic enzyme involved in amino acid metabolism, long chain fatty acid uptake, and here you go, everyone, the tricarboxylic acid cycle.
Who doesn't forget that?
Haven't thought about that in years.
Exactly.
TCA.
Oh, the TCA. That's right.
Wow.
Which evolved primarily to torture biochemistry students.
Correct. Correct.
So this enzyme GOT2 binds link RNA ACOD1. Okay, and they showed that. Now
that in itself is a little bit weird because I had thought of link RNAs as being these, gene regulatory RNAs. They bind to their cognate sequences and inhibit genes at one level or another. And this thing's binding to a protein.
Yeah,
I don't know.
Yeah.
That seemed like news to me.
They confirmed this interaction and they showed that it happens in the cytoplasm.
Yeah.
Not in mitochondria, which is where a lot of these metabolic things go on, right?
Yep.
Right.
You have something to say about that, Rich,
they show the RNA is in the cytoplasm.
Yes.
Right? The link RNA is in the cytoplasm. But there's two major forms of this enzyme, GOT1 and GOT2. GOT1 is cytoplasmic. GOT2 is mitochondrial. So that, to me, is a disconnect.
Right. GOT2 is the one they identified as interacting, right?
That's right. So they've got what, according to literature, is a mitochondrial enzyme interacting with an RNA that they find, at least predominantly by an immunofluorescence signal, in the cytoplasm. Mm-hmm. And they don't comment on that. So that's
true. That's true. Now, this is a cool experiment. They knock down GOT2, reduces VSV replication.
Yeah.
It's just like knocking down the link RNA did. And they called it, this is a nice word for this, phenocopying, right?
Right.
Either manipulation, knocking down GOT2 or knocking down link RNA ACOD reduces VSV replication. And if you overproduce link RNA ACOD1, what do you think would happen? You could predict.
Virus.
That's right. Dickson got it. Promotes viral replication.
Stop pandering to the lowest common denominator.
No, you're the only one who said it. It's good.
Hey, who picked the article?
You did. Actually, Dickson picked this article.
Is that right? Cool. I was wondering where he came up with this. Good.
He comes in the morning and puts papers on my desk, actual physical papers.
That's right.
And
this was behind a paywall, but we got around it.
You get a subscription to the journal, right?
Do.
That's it. Right.
Well, good pick, Dickson.
Thanks.
Yeah.
Thank you for interpreting.
Did you look at the next paper? Yeah.
What's the next
Which
paper?
I think you guys need to know for your textbook.
What's that?
Architecture of Eukaryotic mRNA 3'N processing machinery.
Yeah.
From Laurie Passmore's lab at the MRC in Cambridge. First author is also a woman.
Right.
That's the one that followed this one?
Yep.
How nice. I'm not sure I got to that one.
So the idea here is that link RNA is binding GOT2, and that somehow is turning up metabolism to facilitate virus replication. So this enzyme catalyzes reversible transamination between metabolites, such as oxaloacetic acid and L-glutamic acid into L-aspartate and alpha-ketoglutarate. Rich, you investigated this a little. So can you tell us what would be going on here?
Well, a bunch of stuff. I mean, once again, a good conscious virus over millions of years of evolution knows how to find the key points in metabolism to mess with. And it looks to me like this is one. So first of all, as you also point out here, Vincent, glutamic acid, can be an important carbon source. Mm-hmm. And in fact, in part of my reading says in brain, it may even be a more important carbon source than glucose. By carbon source, I mean this is some sort of carbon-containing molecule which gets broken down in the cells, and as a result, energy is generated. Okay, so glucose, the number one pathway is glucose gets broken down to pyruvate, and you through a bunch of steps, you make a bunch of energy in the form of ATP in the process. So glutamate is another potential carbon source, and the first step in that involves this enzyme that removes an amino group from glutamate and transfers it to oxaloacetate to make aspartate. That's why they call it a transamination. It's moving an amino group from one molecule to another. But the byproduct of that is alpha-ketoglutarate, which is a TCA cycle intermediate, and you just churn away from that, and you wind up with pyruvate out the other end and energy, or in fact, fatty acids. So glutamate is a carbon source for both the production of energy and for fatty acids. But there's another really interesting process that if I ever knew it, I didn't know it for long and probably forgot it, which is the malate-aspartate shuttle. Now, this is really uber stuff here. And I got a little picture, and I probably can't describe it adequately in words, but I'll try and summarize. In order for energy production, you through glycolysis produce NADH, which is effectively a proton source. And you need to get that into the mitochondria to then build a proton gradient that drives the ATP synthase. Okay. The problem is that NADH won't go across the mitochondrial membrane. Mm-hmm. And so what the deities decided to do was to take the electrons from NADH and transfer them to another molecule, which is malate, in a reaction that converts oxaloacetate into malate, and then import the malate and then reconvert it back into oxaloacetate and regenerate the NADH inside the mitochondria. So now you got it there to do the energy production. It's just a funny way to get the NADH into the mitochondria. But now you got a bunch of oxaloacetate into mitochondria. What are you going to do with that?
Right.
What you do with that is to use GOT2 to turn that into aspartic acid and pump that back out into the cytoplasm, and then use GOT1 in the cytoplasm to turn that back into oxaloacetate. And so this is the malate-aspartate shuttle, which is this totally Rube Goldberg way of getting NADH into the mitochondria. I love it. So if you mess with this,
it is true.
Okay, you're messing with energy metabolism.
Hey, Rich, did you ever see the movie The Producers?
Yes.
This sounds like a scheme hatched by the producers that actually works.
It's just
where did we go right?
You could take the malate-aspartate shuttle from Times Square to Grand Central.
You could. You could.
So basically, this is a way to turn up energy production and fatty acid production as well.
Yes.
Now, go ahead.
So this enzyme will turn up energy. Yeah, you said it. Energy production and fatty acid production. It's really a pressure point here, I think.
A couple more experiments, as if that weren't enough.
Yes.
All right. They show.
I'm sorry, my mind is full.
That binding of the enzyme. The binding of the enzyme. So this link RNA binds very close to the substrate site on the enzyme, and it enhances the catalytic activity of the enzyme. The binding of this link RNA.
So it catalyzes the catalyzer?
No, it does.
It's basically an allosteric effector.
Yeah, it's an allosteric.
Binds the enzyme and increases the Km.
Yep.
It keeps it open in one direction?
All right.
But it binds near the active site.
Yes.
Mm-hmm.
That just also seems weird.
Well, close enough, I guess, to make some kind of a change that increases. Yeah. But how it works is going to be interesting to sort out. I think that's interesting. I don't know I mean, I'm not aware of, I'm sure there are RNAs binding to enzymes to increase their catalytic activity. Do you have another example of one of those?
I don't have one off the top of my head.
Got to be there.
Must
be obvious.
Yeah, I doubt this is a first for that, but.
Well, right. What about an RNase that just binds and clips an RNA? I mean, that's the binding.
Yeah, I think that's probably it. Yeah. Now, the metabolites have got to aspartate and alpha-ketoglutarate in mice that lack link RNA ACOD1. These are reduced. Okay, that makes perfect sense. And because you're depressing the enzymatic activity if you don't have this link RNA. If you give mice that lack the link RNA ACOD1 gene, if you give them intraperitoneally aspartate or alpha-ketoglutarate before VSV challenge, boom, you get increased viral replication and lethality. So it complements the defects by not having the gene for this link RNA. You supply the metabolites and you get phenocopying, basically. I think that's really cool. That is
the
like,
whole thing is amazing.
And all these are like one sentence results in this. And each sentence is so much work. It's one of those papers.
That last bit says that it basically tells you that the whole point of the virus upregulating this link RNA was to get the metabolites.
Yeah.
Mm-hmm. Mm-hmm.
Mm-hmm. I mean, probably. It's
just beautiful. It's a wonderful idea. And the whole idea that you need to tweak metabolism, and this is why you do it through turning up a link RNA. And, you know, parenthetically, the NF-kappa B activation that does this is probably in response to virus infection, right?
Right.
Mm-hmm.
And it's evolved to say, yeah, you can turn on, you can
bring it on.
Yeah, hit the alarm.
Go ahead. We're going to make more ATP as a consequence. So really cool stuff. I think it's a beautiful paper technically. It's really well done. But the idea that, and they say this represents a new way by which viruses use host metabolic networks for survival. Really interesting. And of course, link RNA ACOD1 could be a target for intervention.
Yes.
And as you say, Nissen. And as you say, Vincent, this is probably not where they were going to start.
Oh, no.
This is just, that's interesting.
Uh-huh.
So this is a multi-institute collaboration, which I think has to be because the metabolomics and the mass spec and all that's probably part of it. But I think that's great. And there are 4 people, as someone said, beautiful, just beautiful. And and really a seminal paper, I think.
Yeah.
Yes.
Would anyone gain anything from looking at RNA viruses to see if there are any sequence homologies between the link RNA here and their genome?
So you could have your own link. You wouldn't have
to
depend on the cell.
Exactly right.
You bet. Why not? And Nels is going to do it as soon as he gets
well, would you please, Nels? No, that's why wouldn't a virus hijack this and then modify it to the right?
I know vaccinia tweaks NF-kappa B, but then it decided that that's maybe not a good idea and has genes to reverse that. So,
yeah, I mean, what's fascinating is every virus needs to tap in, obviously, to the metabolism. And then they evolve to do that in different ways, but then to kind of uncover these sort of pressure points where you can manipulate it and see that independently, pretty cool. For me, the question then, why would the host keep this around?
Yeah.
And so we kind of were talking about that earlier. Yeah. And is there some cryptic advantage to having this that might have a viral connection where the virus doesn't have the upper hand?
So my other question would be, phosphoenopyruvate is an enzyme in the glycolysis pathway before you get into the mitochondria. It's a huge enzyme. I mean, it's enormous. It's got lots of different catalytic sites to it. I'm surprised that, or maybe not surprised, maybe we just haven't looked yet, but I wonder if anything regulates that as well, because that's another crossroads of metabolism.
But I think in the paper, didn't they mention that it was either in the paper or the perspective that went with it, they mentioned that in heart cells, apparently this link RNA is active already, or GOT2 is active already pretty constitutively.
Makes sense, right?
Oh, yeah.
So evolutionarily, I mean, the purpose of this would be to ramp up metabolism, which you need to do in many cellular contexts. And then the virus capitalizes on that to crank up the thermostat, as somebody said, when it comes into the cell, because it needs the metabolic products.
So, Dickson, they identified the part of link RNA that binds got to. It is 165 to 390. So it's not too long, but so it could be in some of the bigger genomes. Sure, would be worth looking.
Especially
for.
the mimivirus.
Yeah. So this paper shows why you should remember the TCA cycle and all of your metabolism that you learned and you never liked and forgot.
And how many molecules of ATP did you get?
This paper shows that you can look that up and refer to it when you need it. Yes.
Exactly.
You have to understand it, right?
You have to understand what it's doing.
Because if you look at a pathway, you're not necessarily going to understand
Biochemistry never goes out of date.
But, this happened also, I think, 15 years ago. Some virologists started to get wind of this effect of viral infection on metabolism. I remember a talk by, I can't remember his name, but he ended the talk by saying, this is why you have to remember your metabolism, your Krebs cycle, because it's going to come back over and over again.
Sure.
Cool paper.
Very cool paper.
Thank you, Dickson. Despommier.
Hey.
Now, why did you think this was cool? Tell me. I'm just curious.
Well, because I did remember my old biochemistry. I saw some familiar words, and I said, and they had a reader's summary, all right, and the summary was in the beginning of the magazine, of course, and there was no details. Just I said, wow, finally something here can sink your teeth into, because now you can see a direct. I wouldn't say link because that's a pun on the words, but
right.
Connection between the viral infection, the host metabolism, and viral production. And I thought it was good. So I liked it for that reason. I still like it for that reason.
No, it's a great paper. It's very good. Mm-hmm. I mean, as soon as you showed it to me.
Yeah, you jumped on it. I
totally
noticed. That's the first time you've ever done that.
No, it's not. I've done it before. Okay, let's go. Let's do some picks.
Oh, good.
I think my wife is baking brownies.
Ooh. You want to share those? You want to send some through the
smell is wafting in here.
There's got to be a way to do this virtually.
Are they Alice B. Tokless brownies?
No, I don't think so.
Soon they could be legally.
Yeah, that's right. Nels, what do you got for us?
Yeah, so my pick is I'm doubling down here in
this
podcasting festival. So if you listen to TWiV 27, this will sound eerily familiar. But so this is a blog from a couple of professors at Stonehill College, which is just outside of Boston. And this is Brownwin Bleaky and Nick Block. And for their class of undergrads, part of their project is to do a podcast on evolution. And so if you go to the link, you can hear the fruits of this labor of the undergrads, some really punchy, short, spicy podcasts.
Spicy?
On,
he likes that word.
Interested, it's one of my, on kind of doing what we do. And I thought it was pretty inspirational and sort of, in a sense, a relief that if any of us went out with a bad voice or pulled a larynx or whatever, there's plenty of
we don't pull our punches. That's the only thing we don't do.
Plenty of backup talent waiting in the wings. The next generation of science podcasters, perhaps right in front of us here.
Yeah, that was nice to see. And if you search, you can find a lot of things like that. Having it associated with the class is pretty cool. I like that. Thank you, Nels. Alan, what do you have?
I have, let's see, which one did I pick? Oh, yes. Place your bets, ladies and gentlemen, for when the sky is going to fall. Okay, just a little tiny piece of the sky. This is China's space lab. It has gone into a decaying orbit, weighs about 9 tons, and there's nobody on it. They're all done using it. So this is the Tiangong-1 space lab. It's been up there doing missions for a while, and it is going to come down. So it's not likely to hurt anybody. It's going to disintegrate in the upper atmosphere. They've gotten, some of the stuff off, and it doesn't seem like it's going to do any damage. But just for fun, these folks have set up a poll or a pool where you can go ahead and go to the Aerospace Corporation, and they are involved in this somehow. I think they may have made some parts for it or something. Anyway, you can put in your email and your name and pick a date for it to enter and a time. It's going to be sometime in March, most likely. And you can pick it. And if you win, they'll send you some swag, I don't know, T-shirts or something.
You don't have to do a crossword puzzle.
You don't have to do a crossword puzzle. You can enter to win. Anybody can enter this thing. And you have to be at least over the age of 13. And I think you're signing up for their news list or something. But I thought it was kind of a cute idea.
It could make a nice display in the sky if it happens at night, right?
Yes, it could. It could be a little fireball up there. And I don't know. They're tracking it obviously very closely to get a trajectory on where it's going to hit. Most likely an ocean.
So they say it has hydrazine. Of course, that's rocket fuel, right?
Yeah.
Really?
They say don't touch it if you find it on the ground, and don't inhale the vapors.
So if you see a fireball in the sky and then some smoking wreckage on the ground, don't run up and grab it.
Yeah.
Okay, Dickson?
Just as a general rule.
It's fine.
Don't go grabbing it. Okay.
Don't go grab it.
So do we have any rough idea, like within the next X years?
March. March.
Oh, sometime in March.
Mid-March is the forecast on it.
Cool.
Yeah.
I didn't realize they had something that was coming down, but there you go.
Didn't you see Gravity?
Well, they had a crash. That's why that came down, right?
She jumped into the Chinese.
But she took it down.
She did
herself. Yeah. She re-entered it, right? Yeah. She fired boosters to slow it down.
She did.
Yeah. Oh yeah, that was great. I like that movie. Yeah.
Excellent. It was just the escape pod, not
made me glad I'm not in space. Rich, what do you have?
Well, I have had on my pick list for a long time because I wanted to try and understand it, Oxford nanopore technology. And after I looked at Nels's bioarchive paper, I thought, what a great opportunity. I figure Nels, having used this, and Alan, I think you've written about DNA sequencing technologies, might be able to help me out with this. At any rate, so I have a nested pick. It's the Oxford nanopore home site, but I was looking around for a video that would explain this, okay? And it turns out that Oxford nanopore actually has a YouTube channel, so I linked that. And if you look in the YouTube channel, you can find a short video on the general technology of the Oxford nanopore, and one more specifically on DNA sequencing. And I've linked both of those. And then one other that's a talk by some guy who I think visited Oxford nanopore and gave a talk on what he calls the sequencing singularity. And I've not looked at that whole thing, but it looks pretty interesting. It's the idea is that sequencing has become so powerful that it's a frontline tool for doing all kinds of stuff. And he, in that talk, actually features work that they did. They were in Africa during the Ebola crisis, sequencing things as they turned up. At any rate, my understanding is that this technology is protein channels in what must be lipid or some kind of impermeable bilayer across which you set up a potential difference, a charge, so that you get the charge streaming through these channels, and then you put stuff on one side of the channel, and as it goes through the channel, it disturbs the flux of electrons or ions through that channel in a way that can be measured and give you information about what's going through the channel. And in the DNA sequencing application, they actually have a processive enzyme that attaches to the DNA and looks like, according to the cartoon, unwinds it as it pumps one strand through the channel. And the disturbance in the field in the channel is related to which base is going through at any given moment, and you get a sequence out the other end. That's crazy. Now, it's a pretty general sort of video that gives you an idea, but I have about 10 to the 8th questions. Can either Nels or Alan add anything to that? Like
Nels has got it.
They have your paper here, by the way, on their website.
Nels has actually used this, so
no, it's been really fun. It's actually been processed for a few years, and what's really interesting is actually to see the growth or the modifications or the development of the technology, even as we've been trying to use it for.
Did you know they have your paper on the front page of the Oxford nanopore website?
Another good reason to post on the bioarchive. You can
another satisfied customer.
There we go. Yeah, I feel like I should be getting some money, some
That's right.
Some free sequencers or something from this. But no, it's really interesting. So when we started a few years ago, first of all, maybe 1 out of 5 devices would work at all. The flow cell. I mean, it was really kind of beta testing zone. literally. And then, but what's happened is it has made leaps and bounds. And so now it's probably more like 9 out of 10 of the flow cells will work. If you have a bad one, you might have to send it back and have it replaced out. But then they've been swapping in and out different enzymes for the channel that you're describing, Rich, and tweaking the chemistry and how you build the DNA libraries. And this has led to several really big advances, and not only it being more reliable, but the accuracy of the reads has gone up. It still falls pretty short for a lot of applications you'd like to do, and you can get around some of that with computational approaches or depth, but that will be something that I hope they can. I'm sure they're working super hard to continue.
You said you're up to 100,000 bases.
Well, so the length is getting longer as well, but the accuracy. So if you're calling the bases, if it's calling junk, then I don't care how long it is.
That's true. Yeah, I can do that.
Yeah, right. We can make our own company today, this afternoon.
I've got one for you. And a bridge to sell you in Brooklyn.
There we go. Yeah. So I think the last I heard, so we've done 100 KB. The record holder currently is like a megabase in a single read. Now, chocked full of some errors along the way. But as that improves, it's really fun to see that kind of change and see how it we come closer, I guess, to the singularity as it was proposed in that last link. So that's, it's pretty fun. And what I love,
you have to,
go ahead.
I'm sorry. Go ahead.
Well, I was just going to say that, so that's these flow cells. Those are the disposable devices that fit in this sort of harmonica sized shell. And that's how that all, then
that's the membrane. That's the membrane and the protein and stuff, right?
Correct. Yep. And you just squirt a little bit of liquid on top of it, which is your DNA library, and then you go from there.
What do you do to the DNA before you add it?
Yeah, great question. So it's a little bit like building a genomic library using other technology. So it's very close to the Illumina, but you might play around with whether you fragment the samples to get shorter reads or to pick the length that you want. Or if you're really delicate with your sample to try to keep them as long as possible, you don't want to break them. The other thing that's coming online, in sort of in the beta testing phase, the way the DNA was a few years ago, is direct RNA sequencing. I am particularly excited about the possibility of this, thinking about viral transcripts and how they might be modified in sort of form and function depending on mutations in viruses or differences in environments of the infected cell. So I think there's going to be many more cool things that we can only sort of imagine in the pipeline.
So one of the things that I have the biggest problem with here is that I can kind of sort of imagine how from one channel, watching the fluctuation in signal from that channel, you might get a sequence. But you must have in a flow cell, a slug of these channels all firing at the same time. I would think unless you somehow can look at each one individually, the average of that would just be noise. Am I thinking about this right?
100% right. Yeah. And so you're right. You're running potentially hundreds of channels at once. You can actually, there's a little on the graphical interface for the software as this is running in real time. You can see whether each channel has a little report and a color code for whether it's being occupied by DNA, whether the DNA is threading through, and if it's giving you productive sequence back. And so
you are getting reports from individual channels.
Correct. And so it has that resolution, or each channel is insulated enough that you can separate out those electric signals independently. And then it sends it. It used to, this is kind of changing in real time as well. So it used to be that it would take all of the data. It's not that much data, but it's these independent streams of data that is basically interpreting that electrical disruption caused by the DNA base as it filters through. Send it up to the cloud where they have proprietary software to then decode what the signals are to the base pairs and then send it back down to your computer. Yep. So they've now changed, so you can just do this all on your laptop. So you just plug it in with a USB, it sends it to your own processor, and they've sort of let go of some of the software, and then it gives it right back to you.
It says here they have 512 channels per minion.
Yeah, the Minion. Yep. And so
I like Minion.
Minion.
It's a one-eyed monster.
I think you might like this, Alan. We've named our Minion devices Iron Maiden and LeBrian James are our two.
Nice.
Yeah.
Like it.
Yep. But yeah, so the all that processing is happening real time. And each one is sort of unique. So, and you rarely, if ever, will see all 512 channels. You might get like 150 that are active, and those can kind of change throughout the course of the experiment. That can last about 24 hours.
And if I'm not mistaken, you use a Drobo to put this data on, right?
That's right. Our old sponsor of TWiV.
One of the Drobos we got
data back
way back when we were supported by Drobo. Nels uses for his minions.
Our hard drives are filling up with more and more vaccinia data.
Also, to follow up on Rich's conundrums, when you set the sequencer up to begin with, can you initiate all of the DNA molecules to go through at the same time?
Yeah, so you actually, right when you start, before you put your sample, there's sort of a testing zone where you just squirt a little something to sort of prime it and get it going. And so it's, the hypothetical hope is that as many of them kick on actively simultaneously. In practice, you don't ever get to that sort of optimum.
And so maybe their electronics has a way of sorting just those out to read for the sequence.
Correct. Oh, yes. Yeah. So if it detects that if it's either a dead channel, and remember, these are enzymes, right? And so and so if there's a dead channel or a jammed channel, then it just goes off, and then that kind of simplifies your computational.
I'm imagining that the pores themselves, the enzymes that are make the channels, are like totally fabricated. Correct. They're using bits and pieces from Mother Nature and putting them together in a way that suits their purpose. That's exactly right. On basically a nano scaffold for each nanopore. Yeah,
it's amazing.
Yep. Amazing.
Processor that are in your computer. That's amazing, but this is
beyond amazing.
This is equally amazing. Because all the things that are happening in the technology is just great.
Yeah.
And I'm sure
I'm glad I pulled this out now, because I really needed help with this.
It's just great. Now,
thanks,
I just
Rich.
wish I had money to do this, because I can think of so many things.
Mm-hmm. Really fun.
All right, thanks, Rich. Dixon, what do you have?
Well, I'm back to my old Cosmos magazine, but I think you'll find this fascinating. And this is a shout out to Nels, who I didn't even know you were here today. So this is totally independent of your visit, but I'm glad that you're here to see this. And this is a description of 18 new species of spiders that look exactly like pelicans. And they were found on, of all places, Madagascar. So I've listed the article in Cosmos, but I've also, as Rich would say, I've nested the actual article within this, so you can actually check out the whole article itself. And at the end of the whole article itself, it has a series of better pictures of the spider. This is in profile. It looks exactly like a pelican. And nature has no bounds. So, Alan, I have a question for you. What do you think these spiders feed on?
I'm going to guess spiders.
No. They feed on silverfish, just like pelicans.
I was going to guess fish.
How big are these guys? They're small, huh?
They're small. They are small. So they were missed. I mean, they haven't even been seen until very recently, so they must be very small. But that was just a joke, by the way. Silverfish would probably be the right food for these things. But they are astoundingly different from most other spiders. And so I would say, wow. Just wow.
Cool. We should sequence it.
Hey,
super.
You could do that.
Let's
do
it.
That.
Let's get
wouldn't be wrong.
That's right. The forms that emerge, the diversity of forms and morphology.
No question.
Oh my goodness.
And spiders are sort of marginalized.
They are most wondrous and varied.
They are.
But
they are, Chuck.
And if you look up industrial applications of spider silk, they have a new set of Nike running shoes made out of spider silk.
You do?
No, they do.
Oh.
And they're incredibly lightweight, and they're incredibly strong. And, that's the latest running equipment.
Can we stabilize vaccines with them?
Maybe.
With spider silk? Because you can do it with other silk.
I don't know.
Cool.
That's a great question.
Wow.
This Cosmos looks interesting, Dickson.
It's a very interesting magazine.
Because I look at some other articles here. A new archaic species of human. New dinosaur species had tiny arms like T-Rex.
Right.
Bizarre new species of giant shipworm in the Philippines.
There you go.
Yes, I could get into this.
Cosmos it's a.
But no, I'm not going to. This is your thing.
No, please.
Kathy, what do you have?
Oh, I just have to say I've downloaded the PDF of the original article, and Dixon's right. They have pictures of every single one of these spiders. They are amazing.
They're incredible.
I picked something that's very short. It's an 11-second GIF that shows you how taking a piece of circular paper, put a dot on it, and you fold the circumference over to the dot in many places, then eventually each of the folds will define an ellipse.
It's amazing. It's very cool.
Just something short and fun.
Yeah.
Neat.
So if you just describe it without showing it, is that origami?
Yeah, it is.
All right. My pick is I really picked this for Alan because I thought he would appreciate it.
Yes.
How to take a picture of a stealth bomber over the Rose Bowl. So this is done by a photographer flying his Cessna 206, who he's done this for years. Okay. It's not like he decided to take a picture of the Rose Bowl, which was recent, I guess, right? Beginning of January.
Yeah.
And he flew over it, and a stealth bomber was under him, and he has a picture of this bomber over the stadium, which is full of people, and it looks like it's photoshopped, but he says, no, I actually took that, and it took a lot of planning to do it, because he's going pretty fast in one direction, and the bomber's going pretty fast in the other, right?
100 miles an hour, 200 miles an hour.
Yeah.
Yeah.
Things happen quickly. So he talks about how he did it. So I thought you'd like this, Alan.
And he gets it so that the
cool.
stealth bomber is not covering up the word Oklahoma or Georgia or the flag.
That's right.
It's just amazing. January 1st.
January 1st was the Rose Bowl.
Mm-hmm.
Yeah. We have a listener pick from Gretchen.
And I guess this
Yes.
B2 is probably on final approach for something, or
I don't know why it's there.
It's especially going over because it's the Rose Bowl.
Rose Bowl.
They have flights go over.
Oh,
Yeah.
it's going.
I was at a Rose Bowl once, and they have a flight
doing a
come
flyover. Okay.
Yep.
Yeah, my wife saw one of these. We've got two major air bases near here, one of which is Westover. They fly huge military jets in and out of there all the time. Those are mostly C-5s. But my wife came home one evening and she said, I saw the weirdest thing fly over. And she was coming home from right in that area.
Right.
She described it to me. I said, oh, that's probably the B-2 stealth bomber. And she said, no, there was nothing stealth about this thing. On radar, it's stealthy.
Right. That's right.
Well,
Yeah, it looks like a UFO.
Actually, we saw one fly up the Hudson River once.
You did?
I did. I looked out my window. I said, what the hell is... Oh, I know what that is.
Neat.
Yeah.
We have a listener pick from Gretchen. I recently visited the Cleveland Museum of Natural History and encountered an exhibit there that I thought you would all like to know about. And this is called the Bearded Lady Project. Women have made discoveries since the beginning of scientific inquiry, yet are often less celebrated or even acknowledged than their male peers. This national touring photography exhibit asks the questions, would these women be better known if they had a beard? There you go.
Oh.
It's a traveling exhibit. Thank you, Gretchen.
Cute.
It's cool. Yeah. It looks like, I can't tell from all the names, but at least some of the curators are women.
All right, that's TWiV. You can find it at Apple Podcasts, microbe.tv/TWiV. Send your questions and comments to TWiV at microbe.tv and consider supporting us financially. Go to microbe.tv/contribute. You know, at the end of last year, I put out a plea for support, and many, many, many people have responded. Thank you so much. We really appreciate it. Nels Elde, he's at selvolution.org. He's Early Bird on Twitter. Thanks, Nels.
Hey, great to be here in vivo in the company of friends and podcasters. I can't think of a more fun afternoon.
Let's do this again.
Dickson Despommier is at parasiteswithoutborders.com and most importantly, thelivingriver.org. Thank you, Dickson.
You're welcome, Vincent.
Thank you for the paper. It was very nice. I expect one regularly now.
Roger that.
Roger that. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Rich Condit is an emeritus professor at the University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich. He's gone.
Oh.
He's probably muted.
Yeah, I was muted.
What do you have some visitor in your room?
No, I'm still hacking up debris from my virus infection. So
I thought maybe you were having brownies.
I thought he was stuffing brownies in his face.
Stuffing brownies.
It's just normal for Austin, by the way, Rich. Get used to it.
So
I said thank you, Rich.
Yes, sure thing. Always a good time. That's my line. Thanks.
This.
Yeah. And great to talk to you, Nels.
You
It's
too,
good
Rich. Always love it.
Alan Dove is at turbidplaque.com. He's on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
And next year, I'd like to see your photo over the Rose Bowl, okay?
I'll see what I can do.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiV and the introductory music by Ronald Jenkins, ronaldjenkins.com. We've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.